Hi, folks. If you're tuning in and hoping to hear from Dr. Needle about bird flu, you're in the right place. Thank you guys for showing up today. If you would like to put in the chat, if you could put your name, well, no, you don't need to put your name, maybe where you're zooming in from. And also, since we're talking about bird flu, maybe put in your favorite bird. How about that? Hope that your favorite bird doesn't succumb or get sick from bird flu. So where you're zooming in from and a favorite bird. And we'll just wait for maybe one more minute before we get started today. Peterborough, she loves the chickadee. Oh, Lisa, I love the chickadee too. Such a friendly bird. Great. That's good. I just saw that Phil Brown is here. And Phil Brown is the director of bird conservation for the Harris Center. So I'm glad, Phil, that you're here. And for those of you who are just zooming in now, if you want to put in the chat where you're zooming in from and a favorite bird. Oh, John likes to see the Swanson's thrush. Well, that is a nice bird. I like to hear it. Great. All right. I think we'll get started since it's 5:30-ish. And let's be prompt and tidy on time. I'm Susie Spickle, Community Programs Director for the Harris Center, and I want to welcome everybody tonight to this event about bird flu, which is an important topic and something that we're going to learn a lot about. Before I talk about bird flu, I do want to give you another program that you can come see. And actually, I see the presenter here in the audience, Miss Emily Rubel, retired teacher. She's going to be presenting on Wednesday, November 19th from 12 to 1 p.m. via Zoom. It's a lunch and learn. So pack your lunch and eat it while you listen to this incredible talk about mighty monarchs. Emily, I'll ask you to unmute if you want to say anything. Can you say?
I'll just say it was a trip of a lifetime to go see the monarchs in the winter in Mexico in 2024. And my good friend Judy and I are looking forward to sharing about it.
That's great. Thanks. So sign up for that if you haven't already. It's going to be a lot of fun. Tonight, we are really, really lucky to have Dr. Needle with us. He was here before in the past. Dave, what did you talk about last time? Do you remember? It was
Fish.
Fisher. It broke like a Zoom thing. We had so many people interested in this topic. It was on Fisher and rodenticide, and it was so well presented and so informative that I said, Dave, what else can you talk about? And then bird flu kind of came in. And so we're excited tonight to have Dave here. He is the senior veterinary pathologist at the New Hampshire Veterinary Diagnostic Laboratory, and he also does some teaching and continues doing research. So really lucky this evening. Thank you, Dave, for being here. And you can, oh, I know what I wanted to let everybody know. If you have any questions tonight, just put it in the chat and we'll take questions at the end. So Dave, take it away.
Can everybody hear me well? You go ahead and minimize the picture of me. I'm a better thinker than looker. I guess the option's open. So I'll give you a brief introduction to me and a very brief introduction to the respiratory tract, just because I think it's really important when talking about influenza. Then we'll talk about influenza virus itself, and then we'll get into avian influenza. So I was born in New Jersey. I went to Tufts. I was a history major undergraduate because history classes were afternoon and not on Fridays. The irony being now I teach a Monday, Wednesday, Friday, 8 a.m. class. And the years that it does happen, teaching on the 6th of May after Cinco de Mayo in the morning is pretty funny. I met a girl and I followed her to Colorado. And then we moved back. We lived in New Jersey. She's on the other end of my wedding ring, and I took all the sciences for vet school at Columbia while I was working in a vet hospital and as a docent at the New York Aquarium. I went to vet school at Tufts because I didn't think they had quite enough of my money from undergraduate. So I was like, here's $100,000 more. So that's me with younger plumage. And if I had not gotten a pathology residency, I had a job lined up to go work in the Northeast Kingdom and play with dairy cattle, which I really do love. But I did get a residency at the Michigan State, was called the Diagnostic Center for Population Animal Health, and they changed it because it was too many letters, I think, when they got audited. So now it's the VDL. My residency was completed in 2014. In 2012, my wife gave birth to Sammy. He's he's this one. This is Delilah, who was my Spay Lab Pitty, who lived to almost 17. And just recently, we buried her in her yard. And so she taught Sam how to chew sticks. If you look closely, he's chewing sticks because she's chewing sticks. And that's my parenting philosophy, hands off. I did a postdoc fellowship in mycobacterium tuberculosis and the year after my residency, and then a job opened up here at the NHVDL at UNH, and I plan to stay here until the end of my time. It's an Oregon Trail picture. I really like it. And then since we've been here, young Zachary joined the crew, and even though he looks really sweet, I torture him. And if you take a look at how happy everyone is, and then look at Zachary's like, what is going on? So, you know, we're raising him to be a tough northern boy. Aside from my diagnostic duty, which consists of biopsies and postmortems, I have a research group that sprang up out of having a conversation with the director of the diagnostic lab about the types of bacteria that cause mastitis in dairy cattle and how they're very different in conventional and conventional and organic farms. And so I had a hypothesis about why that was happening, and I wrote my first grant, and it got funded by the USDA. And I was like, oh, cool, now I'm in big trouble. So since that time, we've continued to basically pursue disease incidences through the lens of genomic analysis and understanding the relatedness of emerging diseases to previous incidents of the same disease or similar diseases. And we do a lot of wildlife and emerging disease ecology. So those two arms, the group, how dare I? So this is Megan Muniz. She's actually a local veterinarian. My wife got a dog before my Spay Lab dog died, and he hates me. His name is Chuck. And I've just, after 2 years, been allowed to pet Chuck. So her dog went to the vet, and Megan was the vet, Dr. Muniz, and she was talking to my wife, and my wife played matchmaker because Megan started talking about disease research. And so now she is a research scientist of our group. Kellen Sawyer is a environmental engineer and like a country boy who can do absolutely anything and build stuff and fix stuff. Zeb Antonioli is from Montana, and he's our first PhD student, and little baby Zeb. And then Samantha is our second PhD student, and she was an undergraduate, and she did her thesis with me when she was an undergraduate. And now she's back and she's a PhD student. She has a 5-year-old daughter. It's like the coolest thing ever. So that's the group. And we have a host of undergraduates that come in and out of our lives. And what we're really interested in trying to understand is how do anthropogenic pressures, like climate change, pollution, habitat fragmentation, and globalization, those are the big four, how do they alter and directly impact the environment? And really, how do they impact the microbiome of the environment and of the host? And how does the microbiome perturbation, be it species alterations or genes that become more prevalent within populations, how do those interact with the host? And what impact do they have on immunology? And where do pathogens fit in within this? And it ends up being super broad, which is great because I have attention span of a goldfish. And it's super fun. And so we've seen a little bit, we're all over the place. And this really is, we are a One Health laboratory. We are looking at the confluence of human, animal, and environmental health. And we're really looking at, I mean, while I am concerned about human health and the impacts that what we do will have on people, my training is in as a veterinarian, and my interest lies in farm animal and more largely wild animal health, and that really is ecosystem health in both cases. And it's our impacts that change those more often than not. So right now, our studies that are funded is that mastitis study in cattle. And it's cool. It's in New Hampshire, Vermont, and New York, and it's across the 3 states. It's 12 farms comparing 6 organic and 6 conventional. And then we have it already submitted, and we're waiting for review, a proposal to look at the microbial ecology of chickens in backyard farms, because it's a really important and understudied portion of the rural economy, of rural health, of food chain security, and it's very prevalent and has become more so. And then on the emerging disease ecology, sort of like what happens, and then we scramble after it. So in 2021, when we were all still reeling from the pandemic, there was a large die-off of songbirds. We sequenced the most birds in that die-off. We didn't find a smoking gun, but it was a really informative opportunity to apply the work that we do to a death event. You may have heard we found what believed to be a new component of the canine infectious respiratory disease complex, and we have funding for the American Kennel Club Canine Health Foundation for that. As I presented earlier, myself and Rem Mall, who's actually the person who got me in touch with the Harris Center, who is New Hampshire's like everything camera trap and collard hero. He's a geospatial temporal modeler. We have this really cool grant from Fish and Game and Fish and Wildlife Service to look at fisher root cause mortality and health and ecology. We just completed funding on a USGS grant to look at ticks of wildlife origin. And we have funding that we just completed locally on a really cool toad pathogen. And then we're looking at salmonella and some companion animals. So we really are all over the place. And that's by the design, and also by the fact that we kind of are unique in how we do what we do, and it's a relatively small state. And so it works out really well for me. It's fun. So the respiratory tract has 3 components. There's the upper respiratory tract, which is your nose, mouth, and then like basically everything above your voice box included. The lower respiratory tract is the conducting portion of it, which is your trachea down to your bronchi and bronchioles. And then finally, you have your lungs, where the respiration, the actual gas exchange occurs. There's a lot more we could talk about in this, but I'm skipping through a lot because I want to get to the virus. But I think it's really important we understand how breathing happens and gas exchange happens if we're going to talk about a pneumonia causing pathogen. So the alveoli are the end of the respiratory tree. They're little sacs. And uniquely in these little sacs, you have the endothelial cells, which are the ones that line the blood vessels that live in between the sacs, and the epithelial cells, the type 1 pneumocytes that line the inside of the sacs, which are the alveoli. They share a basement membrane, meaning that it's one of the rare places in the body where these 2 cells are actually like physically and microanatomically almost fused together. And that's because across them we have polar gas molecules diffuse across cells. And that's really hard to do. It needs to be really thin. And that thinness is super duper important, which is why bad pneumonia is bad, because it has to look like this. This is a microscopic picture of the end of the last size conducting airway into the alveoli, and they're super duper thin. This is a red blood cell, right? So that's like 10 micrometers. So this is thinner than a red blood cell, meaning you have to keep it really thin in order to have good gas exchange, which is necessary for life. So the respiratory defenses are various, just like in other organs. So your normal flora, the bacteria that live there, include bacteria, fungi, viruses, protozoa, other things. The mucociliary escalator and mucus we'll touch on. We're going to skip over soluble defense factors because you guys are not taking a test after this, and I don't need to come up with multiple choice questions. That's what those are for. The epithelial barrier is pretty obvious, right? It needs to be intact. And we're going to talk about the macrophages because they're wicked cool, as they say around these parts. We're not going to talk about the neutrophils because I don't want to. So the normal flora is Staphylococcus, Streptococcus, Haemophilus. What's interesting about some of that upper respiratory tract flora is Staphylococcus, Streptococcus, and Haemophilus are very common, significant pathogens of pneumonia down in the lungs. So if you have transposition of your normal upper respiratory tract flora into your lungs at a time when you have something else going on, like a previous virus or lots of stress or whatever, you get pneumonia. So it's always, important to think about that. So the mucociliary escalator, right, is probably undebatably the greatest Christmas movie of all time is Elf. And so the mucociliary escalator is the means by which you swallow your saliva, you breathe, your lungs make mucus. That all comes up. And that comes up to keep the pathogens out and also to help recycle the fluids that your body makes. Something really interesting about the mucus in the air, along the trachea is it was once thought that it was a simple homogeneous layer of mucus that just got like pushed along by the cilia as they beat them. What was really cool is that they found out that it's actually a bilayer, and that the inner part, like next to the cilia, so if you look here on this diagram, the MUC4 and MUC20 are like bristle brushes of mucins, and they tightly anchor onto the cilia and they increase their surface area incredibly, and they make them able to push more of the outer layer, which is really fluid. And so you have two different viscosities and compositions and tenacities and how closely they adhere to the cilia. And so without the MUC4 and MUC20, you wouldn't be able to move effectively and efficiently the mucus up. It's just super cool. I learned that from I think Jack Harkema, who is a pathologist at Michigan State. Like I said, we're going to skip over the ones that aren't as fun. So resident macrophages are really interesting in that throughout your body, many cells have macrophages that live there. And those macrophages all come from the same source. They're from the bone marrow, and they're monocyte lineage. So like they're big cells and they gobble things up and they help direct the inflammatory response. Of all the resident macrophages in the body, the pulmonary alveolar macrophage. So here's your blood vessel. The blood's coming through. Can you guys see my cursor? Do you want me to make it like red or something, or is that good? It's good. Okay. So here's the intravascular circulating monocyte. It begins to differentiate and flatten out, and it leaves the blood vessel and becomes an interstitial macrophage, which already is distinct from the bone marrow and the blood circulating monocyte. And then it gets into the alveolar space. So this is the air space here. This macrophage, the alveolar macrophage, is the most distinct from the bone marrow origin in terms of the genes it's expressed and its biological activity. Which sort of makes sense. You're literally sucking in like pathogens and toxins and weird gases that can burn, right? And then you're like passing them around and breathing them back out. But anyway, it's super cool. There are ways in which these can be manipulated by different pathogens and different medicines. So that's sorry, pardon me. So now we're going to talk about influenza in general. So influenza is an orthomyxovirus. Orthomyxoviruses are RNA viruses. So this is, I like to call this baby the big wheel of death. I'm not cheering for pathogens to develop, but I'm not sad when they do, because, I got to pay the bills. So you have your bacteria, eukarya, and archaea are obviously small here and shrunken down because this is for viruses. And the orthomyxoviruses are RNA viruses, and they, as you can see, through evolutionary time, have evolved far away from the DNA viruses, and they've evolved sort of early on in the evolutionary path. So here's where most of the RNA viruses are. These are DNA viruses. And then in evolutionary time, things that we think of as being old, like archaea, are way more evolved from the origin and root of the viruses. So thinking about RNA viruses specifically, one way that this is a Nature article, and it breaks them into these sort of 5 branches but they're more than 5 if you count them. And so some of these, this branch here is actually non-viruses. It's other mobile genetic elements. So viruses are zombies, right? They're not alive. They don't reproduce. And so the way they're characterized is that they're not living pathogens. So they don't reproduce in a classical way that make themselves. They have to be in a host, and they don't make waste. They're not living, but these are even less living. They're like pieces of genomes that can be passed around between organisms. So then branch 1 is both a combination of phages, which are bacteria, viruses that infect bacteria, and then some other viruses that infect plants. And if you look over here, we have a branch that includes a lot of really impactful viruses that are highly pathogenic and cause significant and severe disease. And they're far from that initial root or branch. So here you have Ebola, rabies, Lassa virus, and then you have the influenza A virus. So influenza, the orthomyxovirus, and it is most closely related to other viruses that cause some significant disease, and it is branched off from bunyaviruses, which are vectored viruses. And that's a interesting switch right there, because orthomyxoviruses are not vector. They are free viruses that can infect from animal to animal. And when you break the orthomyxoviruses down, there's a couple other orthomyxoviruses that are of concern, but here you see there are influenza viruses. And you see they branch off. Well, anytime you're reading a phylogenetic tree, whether it's like this, where it's unrooted and it's a circle, or if it's like the ones that are like rectangular, the distance, the length of this portion of the branch is what really matters. And so this is evolutionary time. So this, whatever branched off here from 1B and made this, it evolved very quickly into these 2 bigger things. And then these viruses here, the arenavirus, the paribunyavirus, they also very quickly evolved. On the flip side, the branch that became influenzas was done for a long time, and then it evolved out later to make B, A, C, and D. So there are 4 types of influenza. A is the most pathogenic. It is the one that causes pandemics at this point. B causes seasonal epidemics. C and D are mild and animal specific, and we don't really hear about them. For all of the influenza viruses, the natural reservoirs are aquatic birds. And you know, the old saying, you don't know it because it was told to me by the person who said it the first time, but it's funny, is that the idea is, you know, that waterfowl are sort of the villains here. They don't even have the decency to let you know that they're carrying these influenza viruses, and they come into your pond and they make your chickens die. So now that we've bored you, I bored you with the things that I find interesting. We're going to talk about the virus itself. So they are enveloped, which means that they absorb the cell membrane of the cell that they've infected when they pop out to become infected. And so envelope viruses are inherently soft for environmental stability because that envelope can desiccate and is more prone to heat or extreme cold. So it has an envelope and it has glycoproteins. And those glycoproteins, the HA protein, hemagglutinin protein, is synonymous with the spike protein of COVID or SARS-CoV-2. It is the protein that will interact with the host cell. It's the purple ice pop here. Neuraminidase is important in the release of the virus from the infected cell. The genome is a single-stranded negative sense RNA genome with 8 segments. So what you need to know is it's an RNA virus and it's got HA on top of it, and HA helps the virus do its thing. the genome is segmented, and this is really cool from a population ecology standpoint. When you think about the virus, it's an RNA virus. So one, for the most part, RNA viruses have a much higher mutation rate than DNA viruses because the RNA polymerase, which is the enzyme that copies the RNA when the virus is reproduced, does not have the ability to error check. So it's like me writing an email, right? Doesn't matter what I typed, it's over, I'm sending it, here we go, you know? I'll tell you if you, this is slightly off color, but when I'm writing a report about skin inflammation, I have at times sent reports that said dermatitis instead of dermatitis. And then our receptionist gets the report and starts laughing and tells me that I did it again. Anyway, so there's a really high mutation rate because of that, but that's not the real reason why influenza is so challenging to create vaccines for. It's antigenic shift, which is reassortment. So if I had influenza A and someone else had influenza B, B, or C, or A, and I get infected with their influenza, I could change whole chromosomes, whole segments out and make a brand new virus with an entire new HA or NA, right? And so then you can have exchange of portions of those genes. And so if you have a situation like right now where HPAI is now endemic within North America and South America, the Atlantic flyway is every fall and every spring, we're getting new influenza come, and everyone meets together in the plains. The birds, not the people, no one wants to go there. So all the birds meet together in the plains, and then they share themselves their influenzas. And when this HPAI becomes highly infected to people, if you think it's hard now with our seasonal flu vaccine generation to hit the mark, this is going to be incredibly difficult because we just have hundreds of thousands of birds that fly together and go all over the world, right? And these viruses can shift to other hosts. So, happy Thursday. The way that the virus replicates within the cell is really interesting. I just think it's a really cute picture. So the hemagglutinin virus attaches to sialic acid, which is just a residue that's on the outside of the cell, just a little molecule. And that allows the virus to go in to the cell. It gets endocytosis, it gets absorbed by the cell. And the virus goes in to the cell, it opens up, and it lets its RNAs out. Its genome goes out into the nucleus. And then those viral RNAs are integrated into the host mechanisms for making proteins. And making proteins is central to what all cells do to do what they do. Transport, communication, digestion, building, protein. So we're always making proteins. And so the viral RNA gets in there, and that means viral proteins are made. Now we have a bunch of viral proteins that are made, and then we also have copies of the RNA, and then they get transported into the cytoplasm as RNAs and proteins do, and they reassemble, and then they bud out. And when they bud out, they assemble in a really amazing way. Their proteins that they need to have on the envelope are assembled onto the cell membrane, and they bud out of the cell. There's really cool pictures of that. And I'm sorry, just right now, I realized I should have had a picture of the virus budding out because they have electron microscopy of it. But you can imagine it. It's like this drawing, but in black and white. What does it do when it's in you, right? So I think this is really interesting. If you want to, go to sleep hard, you read scientific journal articles late at night after you put your kids to bed. You've walked the dogs and done the dishes. And I read this article, and I even stayed awake almost halfway through. It was a really good article. And what it points out is something we should always consider is that in terms of whole body physiology, it's not just the pneumonia that makes the pneumonia bad. It's the impacts that the virus has on other parts of the body, and the dysbiosis in the intestinal tract, so changing of the bacterial composition of the gastrointestinal tract, can cause motility disorder, and it alters your body's capacity in the lungs to have the appropriate response to help fight off the virus. What happens? You get cough, sore throat, and fever, right? When it gets into the epithelium of the respiratory tract, it actually kills those cells, and then it has a strong immune response. And it's your actual innate immune response that causes those symptoms. Locally, and also then in circulation, you get the cytokines of acute inflammation, which are TNF alpha and IL-1, and they go around, and that's where you get your fever. The big problem is not the influenza, which is terrible. That, you know, for most people, severe case of influenza is the sickest you'll be in your life. But the secondary bacterial infections are often what kill you, or someone. Well, wow, this is getting dark. So, those bacteria, that's a classic pathogenesis. Viral infection, secondary bacterial infection. And the stomach ache and the diarrhea that go along with it exacerbate the challenges for your body. Okay, so I thought this was a funny diagram from the CDC. Influenza milestone. It's like very pro-influenza, but I'm not sure if CDC exists anymore. Anyway, so happy Thursday. In 1917, the United States entered World War I, and in 1918, the Spanish flu came, and by 1919, it had circled the globe 3 times. And so the Spanish flu, depicted herein, was the first severe recorded modern pandemic. We didn't even know what a virus, we knew what viruses were theoretically, but they were just considered non-filterable particles. They were too small to be seen. But then in 1930, experimentally, through isolation from the fluid, the cells that were infected, they were able to passage the virus and cause disease. The flu caused another pandemic in 1957. In 1960, we had our first effective vaccinations that were made. In 1968, there was another pandemic. It was an H2N2. In 2009, H1N1 swine flu. If you guys remember, I do, because I got it. I was in veterinary school, and I had to take an examination, and I very overtly had the flu. I was like 103 fever, but the tests, you know, there was very little exception. And so instead of telling me come back when you don't have a fever, one of the buildings was closed for construction, and they had someone put the test in an envelope in an empty room in a building that was in construction. I went and took the test, and then I had to put it in the envelope, spray it down with a disinfectant, and leave it on the desk and spray the desk down. Someone came in 2 hours later to get the test. I got a 92. Anyway, so we should all know about the preventative measures, which is vaccination. It's not great every year. It's still better than it can be. And not only can it keep you from getting sick, if you do get infected, it decreases duration, it decreases severity of disease, it decreases shedding. So it's your duty as if you're safe to be vaccinated, to be vaccinated. It's good for the herd, which is all of us. There are really cool antiviral medications that are pretty darn effective. I've taken azeltamivir as I get the flu every like 4 or 5 years, even with vaccination. And my daddy calls me in the prescription because he loves me. He's a physician. One thing that we're not as good at in this country, but people are a lot better at other places, although I think we're getting a lot better since COVID, is the recognition that like nobody cares if you're at work. If you're sick, stay home, Dave, you know, and wear masks. And it really does make an incredible difference. So on to the subject at hand. Avian influenza. Taking a 3-second water break. So avian influenza is caused, like all pandemic causing influenzas, by an influenza A virus. Waterfowl are the primary reservoir for avian influenzas. Avian influenza are subdivided into low-path avian influenza and high-path avian influenza. H5 and H7 are the only hemagglutinin types that are high-path influenza. What's really cool and interesting about the difference between low path and high path influenza, it's not really everything else in them. It's the way that their hemagglutinin gene is made. In the influenza virus, as it floats around, they go back, the hemagglutinin gene is actually HA naught. That gene is cleaved into HA1 and HA2. That cleavage point, that cleavage is done by the host. And it's done by called trypsin-like serine proteases. So these are incredibly ubiquitous enzymes that your body makes. And they look to cut specifically after two types of amino acids, lysine and arginine. Doesn't matter what they are. The point is they're specific to these sort of specific parts of proteins. And they have a serine residue in the part that does the grabbing and cutting. Right in the middle is a serine. So that's why they're called serine proteases, and they cleave. What makes high path avian influenza able to cause highly pathogenic disease and kill these chickens is that it's HA naught, the site for cleavage, is able to be cleaved all over the chicken's body. And so it gets disease everywhere, and it's severe, and they die. The low path avian influenzas can only be cleaved in the upper respiratory tract and in the very distal end of the intestine. And therefore, the disease is not severe. Pretty ridiculous to think that that little bit is what changes from this is in birds all over the place and nothing happens to we have to kill 1 trillion chickens in 5 years. So generally speaking, these are not pathogenic to mammals and people, but that is less and less true. So low path avian influenza in birds is really not a big deal. Local spread, mild symptoms. High path avian influenza, severe disease, high mortality. Oftentimes, high path avian influenza arises from mutations of low path avian influenza that occurs either in the wildlife or in the poultry. So this is great, right? Mild symptoms, local spread, except birds fly all over the world. And so that's where the issue stems from, is that birds fly. So all subtypes of avian influenza, these are all influenza A's, H1, all H1 through H16 can be low path avian influenza. High path, only H5 and H7. These are, when we say bird flu, it means high path avian influenza. They're relatively uncommon in wild birds. They get into your domesticated poultry and then bad news bears. So biologically, the species of impact are not the wild birds. It is other species that have the same serine proteases, right? And so marine mammals are classically impacted by HPAI. The last HPAI outbreak, we had death in marine mammals on the coast of Maine, New Hampshire, Massachusetts. Humans, pigs, this is the classic sort of triad of non-avian influenzas. There is a canine influenza, was described by Cinda Crawford, who is an incredibly awesome clinician and scientist at the University of Florida. And she led that effort. She's an expert in respiratory disease, especially basically practicing just in respiratory outbreaks in dogs and shelters. I know her from my respiratory work. What's really interesting about the canine influenzas, there's an equine influenza that's the same H8, and it was a monophyletic transfer. It wasn't from lots of reassortment. It was one moment, and it's a sort of an exception for influenza where the dog influenza was able to infect horses by the acquisition of a single mutation. Generally, that's a reassortment that does it, not those point mutations. So mutations drive smaller change, and reassortment drives evolution, right? And this is a part we're afraid of. Generally, when we talk about avian influenza, bird flus that have happened before haven't really killed very many people. So the transmission between animals, what makes it so challenging is that it's a fecal oral transmission among wild birds. Has anyone ever seen how responsible those Canada geese are about where they poop? They only poop on parts of the world where the sun shines. And so good luck keeping fecal oral transmission from waterfowl to be transmitted to other waterfowl. It's impossible, right? So in addition, any log or anything else that stays moist and doesn't get too hot or too cold, it's going to stay there. And there was a big issue with the live markets with the previous bird flu outbreak. And there was a lot of discussion among this. There's a state vet in New York, but then there's also someone who deals specifically with New York City. And there are 3 different boroughs that have live markets. And these are, culturally important, traditional ways in which people are acquiring their food. But live markets can really be huge amplifiers, especially when you have other birds around. And so coastal cities and cities large pigeon populations and seagulls, you're in big trouble. What's been very impactful to my life is that if you have a bird in the containment zone that's positive, you have to depopulate all of the other birds on that farm, and you're not allowed to move any of the birds within a 3-kilometer radius. And then you will have to do additional testing around there. And this is all very costly and time consuming. It takes people and money. So 2014, 2015, I'm not sure if you're aware, there was an HPAI outbreak. And at this time, at the beginning of the outbreak, I was still at Michigan State. 2015 is when I came here. And my primary mentors during my residency were Thomas Patrick Mullaney, who was the acting director of the Diagnostic Center for Population Animal Health at Michigan State, and Dalen Agnew, who's now the department chair. So 2 highfalutin fellows. And Tom, as acting director, was in charge when Michigan State University's lab was servicing our state and neighboring states through what was the single most impactful and expensive animal health incident in the history of the United States. Just under $1 billion of cost in 2 years with response activities, testing, and remuneration for animals that had to be destroyed. At the fairs, people were no longer bringing birds to the fairs. They did the chicken shows in the slaughterhouses. And so the animals were tested and slaughtered on site, because if they were positive, nobody brought their birds back, to their farm and spread the virus. It was so stressful and such a big wear that I think it's part of why Dr. Mullaney retired. And he was super respected, and he's a great guy. We still talk once a month or so. And I think that it was such an impact. He was on the news weekly. I mean, this is a veterinary pathologist, it's not a public speaker. So great, really incredibly large impact. And where we saw it, you and I, every day, was in the price of eggs. And it doesn't seem like a lot, but when I first moved to the state, I was superbly lucky, and I rented a house on Stewart Farm, where Lorraine Merrill, the former commissioner of agriculture, and John, her husband, and Nate, their son, and Judy, their daughter-in-law, and Sammy and Hannah, their grandchildren, were farming. And it was just unbelievable to get to bring a little 3-year-old baby, and we had our second child on the farm. We were so happy there. And we often talked about how, it's eggs and cheese that grow people, right? Eggs and milk. It's dairy, inclusive of eggs, that gives us the capacity to grow populations of healthy, robust, people. And a near doubling in the cost of eggs in a 1-month period of time, 2-month period of time is incredibly impactful to the population. And so that expense is not captured in the nearly the $950 million expense, the expense to all of us, right? And so you know, the impacts go much farther than the very obvious and like tangible ones we can write down. So right now, if you're not aware, which you probably are because you signed up for this Zoom call, and if you're not in the right place, my name is David, and just kidding. I do that every now and then during my lectures because at 8 a.m. people fall asleep. So, is that we are currently undergoing an H5N1 outbreak, and it is severe, and it is long. So in 2020, when we were all ready for another nice old pandemic, not many people heard about it because it has so far killed less than 30 people worldwide. It is a virus of low zoonotic risk. Zoonotic pathogens are ones that can infect people. But a friend of mine who is the wildlife pathologist in Nunavut, in the Maritime provinces in Canada, started having deaths. And they started having so many deaths that if it was a species of bird that they had seen before and diagnosed with HPAI, they weren't even doing postmortems. They were just disposing of them. And then it went to my friend Brian Stevens, who is the pathologist for Ontario and Nunavut, and they got it. And because Canada is filled with such kind people, they shared what they had with us. And it came down the pike, and it came here. And we had a really interesting two different occurrences early on in the outbreak. One, a bunch of Canada geese died on the river in Newington right near us. And that was a pretty big deal because Canada geese don't die from high-path avian influenza, they carry it. So we started to see death in birds that we did not see before. It was also a big deal because the people living on the river there were friends with people who had a lot of influence. And so it was a really big priority because people talked to other people, and we received lots of inquiries about it because the geese were basically washing up on people's front yards. In a small enough state that, you know, everyone's 3 or 2 people of degrees of separation away from someone who works in the government in some way or shape or form. And we're a public health lab. So it was really large. And we were talking with the USDA Wildlife Services people daily about the counts and waiting. And eventually a couple of eagles that were scavenging started to die. What's interesting about that is, while that can happen, it's not a huge thing. But when the outbreak hit Georgia and hit it hard, my friend is a pathologist down there, said that the babies were dying in their nests, the bald eagle babies, because the adults were out scavenging and they got it and they never made it back to the nest. So they lost adults and young stock. And something that was really impactful, and I'm not sure we know the full extent of what happened, is black vultures died as well. Turkey vultures were okay. Black vultures are incidental birds up in our region, more or less, you know. The black vultures are the primary scavenger in the forests of the South. So I think there were probably some changes in the landscape from that. So over time, this strain that clade 2.3.4.4b became dominant, of course, right? You know, and it killed wild birds. It killed wild waterfowl. It even started to kill mammals. And so we saw it in red foxes. The spillover into seals was expected. Spillover to cats was sort of new. And this one is a biggie so far, the spillover into cows. It's been detected in over 80 countries and had continued to increase over time as the pandemic has gone on. Interestingly, from an outlook as to when how quickly the pandemics occurred. This was a 2014-2015 one in blue here. It was a slower burn. And then in commercial flocks, it ripped through commercial flocks in 2021, 2022. It was really impactful. In terms of the cost impacts that the initial pandemic had, while I showed you that near doubling that occurred in 2015, 2020 found us almost twice that peak. And so this is a really large impact in a time when people were really strapped for cash, when we've been out of work, people have been out of work or at decreased work for 2 years. So aside from the animal health impacts, which are really important, right, and the spillover to different wildlife species, it had really big impacts on people in their day-to-day lives. And there were limits. I don't know if it was like that for you, but you could only take one carton of eggs home. And also very frustrating for me in the pandemic because I've been baking bread for a long time. Everyone decided they were going to bake bread and they all just went straight to sourdough and they took all the bread flour. You can't go from nothing to sourdough, and don't waste my bread flour. Take your regular until you get your stuff, worked out. You can use regular flour. It was horrendous. Forget about the toilet paper. That's not important. It's the bread flour that matters. So this has been a really important development, and it's really important because it's a threat to human health, and it's a threat to children and our elder communities. So we said that the HAs hit those sialic acids. Remember I said those are just like little surface molecules. It turns out that the udder of cattle shares sialic acid residues with chicken, pigs, and humans. Pigs are a great mixer for influenza. So not only are dairy cattle getting infected, they are passing it horizontally between each other. It is a host-adapted influenza now that lives in dairy cattle, but they're passing it through their milk. And so they have both avian and human sialic acid, which are the receptors for the HA molecule. So they're infected. It causes some decrease in production, but they also excrete the virus in their milk. And then on top of that, very commonly you have kitty cats and you have mustelids around farms, and they are also susceptible to this virus. So the people that have been infected for the most part have been those that have been at depopulations of infected birds or dairy workers, and they're not wearing PPE for personal protective equipment. And so it's a really high impact, high intensity, exposures that have caused positive cases. I have had multiple positive animals that I have done postmortems on, and I wear a papper because of my beard. So that's one of those like space helmet things that blows the air out. And I've yet to have this virus, and there's a lot of it when it's there. So just like with COVID, when there was that amazing spillover from humans into the farm's mink and then spill back, infecting the people who brought the virus in with a new COVID virus that had circulated throughout the mink farms. We're concerned here. But this is a biggie. It's the first one. So, so far, over 500 million birds have been culled globally. So of the affected birds, 77% have been egg layers, right? Remember that 2014, 2015 was a whopping $950 million. The current outbreak has used over $1.8 billion directly traceable money. The egg shortages and costs, both through shortages and other impacts, have been estimated in the $14 billion range. These economic losses are faked, right? Like that's monopoly money. Just to know that these impacts are so incredibly high, which is why it's really important to maintain the institutional knowledge and personnel for those people who work in the field, following and helping us try to control this virus. From my standpoint, this is really interesting. Polar bear is the first polar bear ever to be reported with HPAI. The cattle, and now it's in many states. Goat as well, in Minnesota. A walrus, which is potentially also some impact of alterations to flyway patterns and such. And then a leopard cat in South Korea. It's a wild leopard cat. It's the first wild leopard cat reported. So this is all over the map, right? You got Ursus, you got a Bos, you got a Capra, you have a pinniped, and you have another Felid. So we're talking about a lot of variability and host infectivity. And it's concerning. It's concerning for wildlife, but it's concerning for us as well. And so I put it on a timeline here, just to give you an idea. And then I crop the word timeline here just to make that idea a little foggy because I want to maintain some mystery so I can be invited back. You know, I don't want to show you everything I know in a logical way. And what's troubling here is the leopard cat it was just this year, right? So I was certain actually we had some encephalitis in some of the fisher from our study. I was like, bing, bang, boom, we got the first fisher with it. It wasn't. So we're still chasing that down, but it was interesting. To peel it back to the cattle, this is so far the total outbreak in cattle. There, number of confirmed cases in cattle by state. You see, like California is one of the 5 most important dairy states in the country. To have that big of an impact there. Plus, if you remember from interactions you may have had or heard that the vast majority of the people that work on dairy farms are underpaid and on these large dairy farms out west, and oftentimes can be migrant workers. And so the possibility for healthcare, let alone PPE, is really diminished. The impacts in Idaho were actually really big. It's a really big impact on a sparsely populated state. So far, knock on wood, we have not had it come here yet. But if it's in North Carolina and Ohio, my money's on it's going to be in Maryland soon. This would be a problem, right? Delmarva, this peninsula is shared by Virginia, Maryland, and Delaware. Is the largest chicken production area in the country by density. Even though it's bovine specific, that's we really don't want that to be there. So globally, there's monitoring that occurs through the World Health Organization, the Foreign Animal Disease Office, and the OIE. Surveillance and control of animal movements is superbly important. Education and control of workers and visitors to farms, production facilities. Oops, sorry about that. And even feed, right? Really interesting, different disease, but there are intestinal viruses that cause coronaviruses that cause diarrhea in pigs. And when a new one evolved, it came to Michigan State when I was there, and it literally was brought in to a university with a pretty controlled farm by someone who was trucking in some feed, and his boots were dirty. And they figured it out. It was really cool, but also not cool. So even in the population of people who work professionally, we really have to think about education. And it's like not how I believe the world should be, but potentially thoughts about siloing what people do. When we get birds in, right now we have 2 people who have significant bird populations at home, and we don't have them take the birds in. We keep them away from that procedure. And then vaccination is a big part of it, and unfortunately culling will be as well. And then increased funding to me specifically, to increase my amount of pizza ingestion. No, just kidding. Increased funding for active surveillance in wildlife and domesticated and farmed animals is a really important thing. A lot of wildlife rehabilitation facilities collect bags and bags of animals that die under their care. And some of them might have subclinical disease of real importance. And I think there's an easy model there for us to keep on top of it with honestly free, samples. So the key takeaways here is that influenza in general is a rapidly evolving virus. This HPAI is specifically treacherous for us in that it's our first experience having endemic high-path avian influenza in our country. It's been 5 years. I don't know if the USDA has called it, but I'm going to call it. It's endemic. So I just probably lost my street cred somewhere in Washington. It's evolving. It evolves rapidly. Vaccination remains key. If you have extra money and you don't want to give it to me for research, my recommendation would be to figure out what companies are doing a really good job developing HPAI vaccines and put your money in there. Because even if it's just bovid and caprid, goats and cattle, but the risk for us is important. So lastly, give you Zachary asking questions, and I welcome your question. That was 59 minutes and 53 seconds.
Well, it's really great, Dave. And I'm 100% sure that people have a lot of questions and already have a few. So I'm going to start with one from Janet, who says that they keep chickens and they're really careful. They change their shoes after they've been in. They have like chicken shoes they wear. They wash their hands. They take in the water so wild bird poop doesn't end up in it. But they're wondering what else could they be doing to protect their chickens and themselves?
Yourselves, I don't think you have to worry. You're not in a position where there's a die-off and you're exposed. So it's really a very low zoonotic potential in that regard. I mean, I'd be more worried honestly, about COVID and our seasonal flu variants right now. So get your vaccinations, if you're feeling sick or someone who's sick, wear a mask or don't see them. For the chickens, the higher risk areas for backyard chickens are usually places where there's a body of water on the farm and it brings in high traffic. It hasn't been a feeder bird disease yet. And so like those sort of localized risks have been low. So I'd say continue doing what you're doing. Don't go dancing like wild bird poop and then bring it to your chickens. I think having chicken shoes is a super cool idea. It's really smart. If you really wanted to go, to the nines, you could look at hosting a foot bath outside of your chicken coop for you, so that when you come in and you put on your chicken coop boots, you step in the foot bath with a disinfectant, and then you go do your chicken work, and then you come out and you leave your boots there. To theoretically work. That might be a step too far, but it's not a terrible idea. But I think they're doing okay. Try to, limit access of wild birds into their coop.
A follow-up question from Esther. And this is perfect follow-up. She's wondering, should we not put up bird feeders until our chickens are confined in the coop by snow?
It's not a feeder bird problem. You shouldn't have bird feeders up yet anyway, because the bears aren't sleepy. So I would follow Fish and Game's recommendation. Is that a daily question? Like we put the feeders up when the chicken are in their coop, or can you restate the question?
I think she's concerned that if they put their bird feeders up, even after, let's say it gets really cold, but there's no snow, could their chickens get infected? And if they have bird feeders up, would it then affect birds that are coming?
I don't think the feeder birds. I don't think passerine birds are really susceptible. The only exception being that we've experienced. There's a orthoreovirus, it's called, that causes winter death in crows. They have these like quick hemorrhagic diarrhea or enteritis, and they just die. And so there was one that happened in Maine, and then the next year, same park, like same place in Maine, and bam, it was HPAI. So that was a surprise. But generally speaking, and then corvids are sort of an exception to what you think of as a feeder bird, but it hasn't been a real impact on feeder birds. And I don't think if your chickens get it, I'm not gonna pass it to the feeder birds. They're gonna die.
Oh,
Perhaps.
yeah.
It burns them out really quickly. Like if you have a flock of 10 birds, by the time, so there was a farm of 50 or so birds. They lost 5. By the time the state vets got there to collect animals, they lost 10. By the time we got the animals, 2 more died that day. We got the positive result. By the time they organized to do the depopulation, they didn't have to.
Wow.
They were all dead.
All
So
right.
it's a really pathogenic in chickens. That and very virulent Newcastle disease will win the award for the fastest killing of a flock of chickens.
Okay, here's an interesting question. Why do you think pigs have largely been spared from this outbreak? And does the idea of pigs joining the HPA1 party in the future, uh, and would it lead to more problems?
Oh, it'd be
Does
awful.
it cause you heartburn, given how pigs and people like to Mr. Potato Head this virus parts and share it? That's the
Yeah,
question.
no, yeah, it would be really bad. And so that's I don't know if you're aware of it's a cool question. Sort of the paradigm of uh, how pigs have become so central in influenza, generation is that there's a classical farming method, used in parts of Southeast Asia where pigs are on the bottom of a stack that, or the middle of a stack that has chickens and pigs and waterfowl. And so pigs are great reassorters. And they have both the ability they're susceptible to multiple different types of influenza. But they don't often get sick, um, en masse. And so, uh, I, I'm like honestly less concerned, but that might just be naivete. Um, but it's the novel ones that I find to be uh, more threatening, both from a conservation standpoint and a public health standpoint. I do work with canine distemper virus, and um, there was sort of a new little subspecies of it that came to Tennessee, and it spilled over from the normal wild mesocarnivores, and it killed some in, zoological collection animals. And zoological collection animals are fun to look at, but they're also very important in conservation. Um, you know, each one can have a really important role in the worldwide conservation of threatened species. So I'm sort of more concerned about those than I am about it getting into pigs. But I mean, that would be bad for sure. Real bad.
Yeah. All right. Just a few more questions. This one's from Phil Brown, who's our director of bird conservation. He's curious, would we expect other wild birds beyond geese, eagles, and other scavengers to experience a die-off? And should we report suspicious bird mortalities?
A, yes, B, yes. So there were snow geese deaths in the plains that every other year before would have been Pasteurella multacida, foul plague. But what ended up happening is it was HPAI. You should 100% report unusual mortalities. You can call if you're a Fish and Game in New Hampshire, Fish and Wildlife in Vermont, Maine Department of Inland Fisheries and Wildlife. You call your state vet. You let them know because this is not a this is a great unifier. This is a concern for all of us. So, suspicious deaths for sure, rapid deaths, birds, just, we're here for that. And those are concerning. So I would anticipate, I mean, we had species that have not died before in birds, die in large numbers. So yeah, I'd be concerned.
Great. All right. Just 2 more questions. This first one's from Jeff. He says, in one of the slides of diseases across the globe, all the countries went from clear green to orange to red, except China, that actually moved back to green. Why might China have seemed to have improved?
I don't know how clean or transparent all data presentation is as far as reporting goes worldwide.
Got ya. Say no
Yeah.
more. We understand.
Yeah, there were similar patterns with reporting of, human pathogen that originated there. So I'm not a conspiracy theorist. We know the origin of SARS-CoV-2 was in bats within a cave. It was reported the sequence of the virus is very close to what COVID-19 is. So that's not the secret. But there were times when it was apparent that there was not as good transparency in data. I think my guess is that is what that is. A lot of open markets, a lot of rural populations with small scale farming, lots of migratory waterfowl intersecting with high populations. I wouldn't believe those numbers.
Got ya. All right, here's the last question. This is from Emily. How has the funding for keeping on top of this been affected by the current administration?
Anyone here going to try to fire me if I answer this honestly?
Nobody's going to fire you from here.
Okay. So they fired all the people who do the testing, and they tried to hire them back like 24 hours later. They literally, in a flash of lunacy and ignorance, fired the people responsible for running the surveillance testing at centralized facilities. Not only that, but there are no hands on deck anymore. So many people at USA have had their jobs cut. Forgetting the number of people, the type of people who have been cut are those that have the most important institutional knowledge. And I'm not conformist. I'm not sure if you could tell. I'm not a big hierarchy person. But there's a difference between hierarchical crap and seniority being seniority and knowledge and experience and wisdom. And another example of how deleterious these actions have been to our safety is there's an individual who ran a nationwide infectious disease eradication program, and that person was retired, and that disease, which is a public health nightmare, is having a resurgence the same time that they let that person go. Being non-specific because I believe it's appropriate, but if you could imagine, right, as bad of a design of a moment for decreasing people in general, but taking away someone who spent 25 years building the national infrastructure and planning and been successful, to be removed in the year when this pathogen is making a surprise recrudescence, it's pretty bad. So we're lucky right now where we are. We don't have the cattle one. Things have been relatively quiet in the birds, but we're always one flyway season away from really having trouble. Thankfully, also, we have relatively low heavy production in our state, but we're not far away from states where there's really big production. I don't approve.
Yes, we hear. Thank you, David. Thank you so much. Thank you for this really informative and such great information this evening. A lot of people are thanking you in the chat, and thanks for coming to all of you for showing up for this. This isn't as light and fun as some of our other talks, but it's so important. So, Dave, thank you, and thanks to all of you. And I guess if you keep chickens, don't go, what was that? Don't go dancing through your chicken coop and then go dancing in the rest of the world, right? And
correct. Don't
yes,
go dancing through goose poop into your chicken coop.
Yes, that sounds right. Keep your
poop and coop separate.
Poops and coops separate. It's a good moral to live by. I would like everybody to keep their eyes on the Harris Center's calendar this spring or early summer. We're going to have David back in person at the Harris Center to talk about an emerging toad problem. So keep your eyes on our calendar for that. And again, thank you, Dave, so much. Such important information and so well presented. Thank you. And thanks. Good night, everybody.
Thank you all.